So let's see now how our B cells mature so they can contribute towards our immune system. B cell maturation, as I mentioned earlier, is antigen independent. Our body, our immune system generates several million different types of B cells. Only B cells which respond to a specific antigen are selected for proliferation. But before that happens, a stem cell, a cell which is going to ultimately give rise to B cell, it divides and forms a pre-B cell. The pre-B cell has the first antibody that is generated on the surface of the cell is IgM. IgM, we'll talk about antibody classes. I know we haven't talked about them, but IgM is a type of antibody. It is the first type of antibody which is produced by a B cell. This antibody appears on the surface of the pre-B cell. We have already said that antibody is made of 4 chains, 2 heavy and 2 light. So the first chains that are made are the heavy chains for the IgM. It's a type of antibody. These heavy chains are called the mu chains. So these are the chains which are first made by the pre-B cell. These mu chains are attached to surrogate light chains. Okay, so remember we have to have our proteins in proper conformation. So in order for these mu chains to have proper conformation, they have to interact with with some type of light chain. Initially, they interact with surrogate light chains. These surrogate light chains substitute for the normal or proper light chains, which happens in the next stage. And when that happens, our pre-B cell becomes an immature naive B cell. So in this state, the B cell has now a proper IgM antibody with its heavy mu chains and 2 light chains. So this is immature B cell. This part of B cell development happens in the bone marrow. Immature naive B cells are released. They go in the circulation and then they go to the lymphoid organs like lymph nodes that we have talked about. So here now B cells produce another type of antibody on their surface, which is IgD. Here I would like to emphasize and re-emphasize that the class of antibodies, we will talk about the classes. Here's IgM, 2 classes are mentioned here, IgM and IgD. A B cell can only produce an antibody with a single antigen binding site, or which can only bind. Antibody has 2 antigen binding sites, but that is constant. If this B cell now decides to produce a different type of antibody, it will still have the same antigenic specificity these particular antigen binding sites have. So B cells move to the lymphoid organs, peripheral lymphoid organs. That is where they mature, and now these B cells can switch classes, make different type of antibodies, or they can stay there till they're activated. This is the first type of cell, which is mature naive B cell, which responds to a particular antigen. And this mature naive B cell is resident of the peripheral lymphoid organ. So when a B cell binds antigen, the antibody on its surface, when it binds the antigen, this antigen basically serves as a ligand. This ligand basically, when it binds this antibody, it signals the B cell. It signals the B cell to proliferate, make many copies of itself, and also mature. Remember I told you mature B cells are called effector B cells. These B cells have a lot of significant amount of endoplasmic reticulum, and their function is to secrete antibodies. Effector B cells are short-lived. These B cells which have become activated, they will produce their antibodies and they will die after a couple of days. Adaptive immune system can respond to millions of different foreign antigens in a very specific way. 2 proteins interacting it is again chemistry and the three-dimensional shape. So the antibody antigen binding site is dependent upon those two things, the three-dimensional shape and the chemistry on the surface of these two molecules. Animals generate randomly a vast diversity of lymphocytes, B cells with antibodies which have different antigenic specificities, and only those B cells will be allowed to divide and mature and form effector B cells which have their antibodies stimulated by binding of an antigen. So it is basically, if you think about it, it is basically the antigen that is selecting the B cell for its activation. B cells can produce the antibodies, which is part of our adaptive immune system. Again, adaptive immune system composed of different lymphocyte clones. So if we have, say for example, out of these 1 billion lymphocytes, B cells, there will be clones of few hundred of B cells which have a specific antigenic specificity. So when an antigen comes in, it interacts or binds the antigen, the antibody, which has specificity for this particular antigen. This binding activates the B cell. It is basically the antibody present on the surface of B cell is acting as a receptor. And you can think of antigen as a growth hormone or a growth factor. That when it binds the B cell, it causes that B cell to divide. Not only that B cell is going to divide, it will also become mature B cell and it will start secreting the antibody. The same antibody specificity that B cell had when that antibody was present on the surface. The antibody specificity is not changing, but now this type of antibody is going to be secreted rather than being displayed on the surface. So what we can say about the immune system, that immune system is ready-made, not made to order. What is the difference? We have a vast variety of B cells, B lymphocytes, which have antibodies on their surface with different antigen specificity. When antigen comes in, we have our B cells are already ready-made. Whichever B cell can bind that antigen will become active, it will divide and it will produce antibodies. If it were made to order, when the antigen comes in, our immune system will have to customize antibodies that can bind to that specific antigen. That would have taken much longer. So our immune system is ready-made and not made to order. Very, very impressive way to neutralize the offending pathogens.